And here we are with Chapter 5, Microbial Metabolism. So the thing to remember about metabolism is that you need a balance between breakdown and synthesis. And metabolism is like a broad umbrella, and underneath the umbrella is catabolism, which are metabolic pathways that are going to take high energy molecules, break them down, release energy and trap it in ATP, and it'll also create a lot of building blocks. And then with anabolism, the organism can take the energy, take the building blocks, and then make whatever it needs. In order to be a very robust, healthy bacteria, you have to do both. You have to get high energy fuel to break down in catabolism, and then once you've broken everything down, you use that energy, use the building blocks to do anabolism. So this is a diagram showing you how ATP, the energy currency or energy fuel of the bacterial cell, it's kind of like a rechargeable battery where you have ATP and that stores all the energy so that you can tap into that energy when you have to make something. But when you use that energy, you don't destroy the ATP. What you do is you break it down into two products, ADP and phosphate, because that's where the high energy bond stores, or that is where the storage of the energy takes place, is between that phosphate and ADP. So then if the bacteria does break down a high energy fuel in catabolism, it's going to release a fair amount of energy. So you're going to take these two startup, ADP and phosphate, and then store that energy in ATP. And then you can use the energy in anabolism, and you're back to, it's like a battery that's run down, ADP and phosphate. And then the battery can charge back up again when you do catabolism and you make more ATP, and so the cycle goes. So the nice thing about ATP is you don't use it up. You don't have to do it from scratch. It's just kind of like a bank. You can put the money in the bank, that's ATP, and then you can withdraw the money, and that's ADP and phosphate. But the bank doesn't go away, and it's still there, able to allow you to deposit and allow you to withdraw. Now, metabolic pathways. The bacterial cell never does anything easy, either do we, meaning that the pathways are very complicated. They're very long, they have lots of enzymes, they have lots of checkpoints, they have lots of feedback, which is what you want, but it does make things complicated. So my job is to try to simplify this for you. So you have these metabolic pathways. Either they're going to break something down, they're going to build something up. And each step is catalyzed by enzymes, because remember, enzymes are proteins that enable chemical reactions, metabolic reactions, to go fast. And that's what bacterial cells want, and they want to be able to do things quite quickly. So the enzymes there are there just to speed up a reaction. And what I mean by that, I mean that the reaction would occur anyways, but much too slowly. So enzymes are there to make sure that you're talking about millisecond time rather than second or minutes. And that can be something that if you don't do it quickly enough, the bacteria would die. So this is, again, just showing you where the high energy bond is storing that energy in AD. So here's ADP, here's the energy, and then you have the phosphate bond is where you deposit the energy. And then you have to break that bond to access that energy. And then when that happens, you're down to ADP and phosphate. Put in energy, you can now make ATP. All right, so the two major types of respiration, and well, the way that we use respiration is you think about breathing. And bacteria are only one cell. They don't have lungs, but they essentially do cell breathing. And so very often aerobic respiration is sometimes called cell respiration, and the whole idea is to conjure up this image of cell breathing. And when you breathe in the atmosphere gases, you're breathing in oxygen and you are breathing out carbon dioxide waste product gas. And the bacteria are going to do the same thing. In aerobic respiration, the bacteria want to take in oxygen, they want to take in glucose, and they want to break down the glucose in a process called aerobic respiration, but they do it in the presence of oxygen. And oxygen can create energy, and that energy is huge. The payback for breaking down glucose in the presence of oxygen is 38 ATP. If the bacteria find themselves, let's say, in water where oxygen is quite low, then they can break down glucose, but they only form 2, not 38, 2 ATP. So with aerobic respiration, you have certain steps that occur. And the first step is glycolysis. As the name implies, it's going to lyse the glucose, break down the glucose. Krebs cycle is next. Krebs is, or was, he died probably about 10 years ago. He was a professor at Brandeis University, and the cycle is named after him. And during that cycle, you generate more building blocks. You also generate a bunch of hydrogens that are then going to enter the electron transport chain and do oxidative phosphorylation. So between the oxygen and the hydrogen pairs, believe it or not, you can generate huge amounts of energy. And so glycolysis in Krebs gives you the building blocks, and oxidative phosphorylation gives you all of the 38 ATP energy. Now, in contrast, because aqueous solutions don't have a lot of oxygen dissolved in it, the bacteria might find themselves in an aerobic environment, and all of a sudden they're in an anaerobic environment. And they can survive, but they will have to consume much more fuel, because without oxygen, they only make 2 ATP when they break down glucose. Now, humankind has taken advantage of this because of what is called a fermentation end product. You don't make a lot of energy, but you make this really unusual fermentation end product that you do not make with aerobic respiration. The end product of aerobic respiration is carbon dioxide gas and water. Woot woot. The end product of fermentation, end product of anaerobic respiration by a bacteria or yeast, yields incredible stuff. It yields ethyl alcohol. So all of your beer and your wine, what do? They take microbes and they put them in grape juice and let them do their anaerobic respiration. And they can then be manipulated into making wine that's 14% ethyl alcohol. You also have fermentation end products that make cheese, that make yogurt, that make miso, and vinegar, and I could go on, but humankind have been able to kind of cash in on anaerobic respiration, and they realize that with their guidance, so to speak, they can end up making not just wine, not just beer, but they can end up making vodka, they can make rum, and so forth and so on. So they can make moonshine at 95% ethanol. So for bacteria, anaerobic respiration can be quite the boon to humankind, and we have figured out how to manipulate the organisms to do that. And that's the end. So I will see you for the next section. Take care.